Structures of DNA. Nucleotides are the building blocks of nucleic acids. They are composed of a phosphate group, a 5-carbon sugar, deoxyribose, and a nitrogenous base. There are 4 types of nucleotides. They are adenine, thymine, guanine, and cytosine. Base pairing rules. Adenine pairs with thymine, guanine pairs with cytosine. Watson and Crick founded this.
Boys,
Base pairs are held together by hydrogen bonds. Between adenine and thymine, there are 2 hydrogens. Between guanine and cytosine, there are 3 hydrogens. DNA is shaped like a double helix. Watson and Crick founded this with using the data that Rosalind Franklin found.
I stopped and started it to go over the parts that I wanted.
5 and 3 ends. They're called 5' and 3', and they go from 3 to 5, but not 5 to 3.
To start off with, the purpose of DNA replication is to pass on genetic material to grow new cells and to pass on the genome to the daughter cell. And DNA replication happens or occurs in late interphase. DNA replication starts at the point of origin, which can be identified by a certain DNA sequence. So then to start, DNA helicase comes in right here and unwinds the DNA strands to separate it. And then once that's done, then there's binding proteins that attach to the strands. Once those are attached, then DNA primase comes in and adds a primer, which is RNA, onto the DNA strands. And once it adds the primer, then DNA polymerase comes in right here. Once that comes in, then it can start adding the appropriate nucleotide to the new strands for the actual replication. The direction of DNA replication is from 3' to 5', and so the DNA polymerase and all of the proteins just follow that line and build off of that. So that is the strand that will always be the template strand. And so there's 2 strands of DNA, and they are antiparallel, which means they don't run in a certain direction. They run 5' to 3' and 3' to 5'. And since the strands are antiparallel, the 5 and the 3', each strand is like the separate, so the 5 is up here, but then the 5 is down here on this one. And so the one that starts with the 5 is the leading strand, because it starts with the 5, and then this is the lagging strand since it starts with the 3. So if we look at this right here, so right here is the leading strand, because it starts with the 5, and then the primer was there, and this is DNA polymerase going, connecting, but then down here is the lagging strand, because it starts with the 3, and so the DNA polymerase can't really work on the lagging strand. And the top strand up here, once that starts up here, is the original strand, or the template strand. And then down here, the one that DNA polymerase is adding on, is the new strand, because he's adding it onto it. So then on the lagging strand, since the DNA polymerase can't do it completely, you get these little strands right here and right here that are Okazaki fragments, which are the short DNA strands that aren't complete yet. And so to fix the Okazaki fragments, DNA ligase comes in, which then it connects these together so it can actually form the new strand of DNA. So then it will come fill in these. So then at the end of DNA replication, you have 2 DNA molecules that are identical, and you get them from just one strand at the start.